this later. Turn my volume up. And I wanted to talk about the reading assigned for this week and also talk about a couple of things that are coming up. And at this point, we're halfway through the course. We're kind of more than halfway through the lectures. We're halfway through the group discussions. And we'll have our midterm coming up the week we come back after the break next week. So I will share my screen. Get the chat back. Okay, before we get going, I wanted to ask you, this is something that where's the polls? I always like to hear from students in person. Okay, so this poll might take a couple of minutes. And if you have an older version of Zoom, you might not be able to do it, but I want to get you to let me know what you thought about the readings that we've done the first half of the course. Which ones did you love? Which ones did you not like or have a lot of trouble understanding? I hope this actually works. I've never done a poll like this before. Okay, I'm seeing something now. All right. It was really nice to see people in person last Friday. It was like so nice to hear from you directly that you liked the paper on wealth transfer or material resources transfer in animals. Okay, so we've got about 2/3 of people have participated. Some people might not be able to if you don't have the latest version, but we'll wait. Let's give it one more minute. Oops. I'm just going to take a screenshot of this too. Oops, that didn't work. Okay, so I'll share the results. Oh yeah, the virus one. Yeah. We read that the week we read the ant paper. So I didn't call it out specifically, but yeah, that's cool. Thanks for sharing that. Same here. Okay, so I'll go ahead and share it with you. Oops, so you should be able to see the results now. Yeah, so interesting. I was hearing from people last week too that the one about the human mating strategies was not a favorite, although a lot of people liked it, but it's kind of the lowest ranked of the ones we've read so far. And people really like the papers about like neat animal behaviors. Oh, and one person, or maybe more than one, didn't like the rock, paper, scissors paper. All right. Great. Okay, so that helps me think about the kinds of papers to use in this course in the future. Okay, so let's go to my slideshow. Okay, so you should still be seeing this. I'm not going to play this video. It's about 5 minutes long, but it's a really neat video about cannibalism in biology. So I wanted to share the link with you. It's kind of an explainer about how cannibalism used to be thought to be this kind of like expression of non-adaptive behavior, kind of like accidents, things that shouldn't happen. But more and more now we're appreciating that it's actually like a real part of the ecology and evolution of a lot of organisms. So it's relevant to the paper we read this week. Okay. So I'm gonna skip through and not play it. Oops. Oops. Okay. So I wanted to start by talking about the kind of business stuff about the midterm and the project, which are coming up after the break. So the midterm is worth 20% of the final grade in the course. You'll write the midterm on Brightspace. It'll be open book, so you can use any resources you want. You just can't work with somebody else. It has to be your own answers. They have to be in your own words. You can't like copy paste material from somewhere else, and you can't distribute the questions and ask someone to answer them for you. Okay, so it'll be short answer questions. The test will be one where you decide when you start writing, and the time window that I've set up is like starting at 8:30 a.m. on Wednesday on March 2nd. So it's about a week and a half from now. And you can start it sort of within the next 24-hour period. So if you want to write it at night, you could do that. Or if you want to write it during our class time, you can do that as well. And then once you start writing it on Brightspace, you'll have 80 minutes to complete. And if anything goes wrong, just get in touch with me and I'll just try to fix it as soon as I can. You know, if something happens and you get booted out, I can just set it up so that you get another chance to redo it. Now, if you're writing it late at night or something, I probably won't get that message or be able to help you until the next day. Okay, so do you have any questions? I also want to give you time today to ask questions about the course material, but do you have questions about the midterm? I don't know exactly how many questions will be on the midterm because I haven't written it yet. I'll write it next week, but I'm guessing maybe like 4 or 5 written answer questions that might take more than just like 1 or 2 sentence answers. There might be more questions than that and then have some like even shorter ones, but I think it'll mostly be kind of a smaller number of questions that will be worth a slightly larger number of points because they will be requiring you to like apply concepts from the course rather than questions that just get you to regurgitate things from the slides. Because it's open book, it's kind of pointless to ask you questions that you can just regurgitate from the slides. I'm not planning, good question, Zainab. I'm not planning to do any multiple choice questions. You're not supposed to be here. Okay, you got to go back up. Okay, sorry. Okay, well, I'll have to help you with that later. Okay, so, okay, sorry.
I want to do it now.
You want to do what?
Send her an email.
Send grandma an email? Oh, okay. Well, I'll have to help you with that later because I don't actually have her email address. So you need to go because I'm in my class now. You have to go, and this is on the recording. Okay.
You know her email address?
I don't, but I know her mommy's phone number. So Ada, please go. Okay.
Her phone number.
Yes, but I can't help you with that right now. So you have to go, or you're going to be in big trouble.
Tell me the phone number.
I can't. I don't have it at the tip of my fingers. I have to go look it up upstairs. Okay. So you're going to have to go upstairs, Ada, or you're in big trouble. Okay. Okay, yeah, I'm not planning on doing multiple choice questions because I just feel like they won't be the best test of your learning for kind of open book. So yeah, but good question. Okay, any other questions about the midterm? Is it strictly on the material from the video? Yeah, good question from Floor. I will include at least some questions on the midterm that will relate to the readings. But I won't ask you, like you can kind of use the assignment, like the group assignment questions as an idea of the types of things I might ask. They will focus on kind of the big picture or like the broad strokes. They won't focus on like, the details of why did they use this type of statistical test or things like that. Okay, so a question about whether there'll be any long answers. So I don't, for this course, I'm not going to ask you any questions that are kind of essay style that require you to write multiple paragraphs. The longest question will probably be the kind of thing where you write one paragraph.
What is that?
Okay, and another good question from Zainab. Are there certain readings we should focus on or all of them? I think I'm more inclined to ask you questions about the studies, not the news articles. So like, you know, the one we read this week, especially because there's no group assignment on it, like that's kind of an obvious one for me to ask something about. Yeah, and then, you know, like things like the papers that we've read that are like peer-reviewed scientific articles, I'm more inclined to pull concepts from those. But I haven't written the midterm yet, so I don't want to be too specific. Oh, okay. Would you have to draw figures in your answers? Yeah, that's a good question. I don't know how to do that in Brightspace. I like those kinds of questions, but they might be tricky in Brightspace. But what I will often do for this course is you'll have an image to analyze. Like you'll be provided with a matrix, a payoff matrix, and you'll have to analyze it, or an image of a social network, and you'll have to answer some questions about it. Like that's a good kind of straightforward test question. Yeah, these are good questions. Okay. Okay, so Neve asks, how many questions will be on the midterm? I don't know yet because I haven't made it yet, but I don't want it to be too long. So I can let you know once I've written the midterm, I can post an announcement saying how many questions, but I'm guessing probably 5 to 10 questions, but probably something like 4 questions that take a little bit longer to answer and to mark. And you know, if there's more questions on top of that, they'll be quite short, like 1-sentence answer questions. But the idea is that you should be able to write it in 80 minutes. And maybe, yeah. Okay. Just trying to think if there's anything else. Oh, I was also wondering, like, because we haven't really had like question answer sessions, I want to give some time for that today, but there is some tricky stuff in the course. So it's kind of, I feel like there are questions out there that just aren't getting asked. And so I was going to ask whether it would help to have kind of a session where I was online to answer your questions right before the midterm. So I was thinking like Tuesday evening or something, like I could be on Discord to answer questions, or if, yeah, or like if it was earlier in the day, I'd want to do it on Zoom. But if it, like, I find that students often don't study until like late anyway. So what I could plan to do is like be online the night before at 9 p.m. or something in case people have questions. So I'll figure that out and I'll send out an announcement later. Okay, great. Okay, so I also just wanted to chat about the project, which happens at the end of the course. And this is just a screenshot from Brightspace. And I just wanted to highlight that I added an updated document with instructions. So when you go to the link down here, it is now, it takes you to like the same file that has all the kind of guidelines for the project, but I updated it this week. So it has some slight edits to it, but same kind of grading breakdown and stuff like that. So please take a look at that, and I'll talk about it a little bit more. And then, oh, I just wanted to also say, like, I'm going to put a link to the midterm in the section above when I have it. You can also get to it from the content part of Brightspace. Okay, so here's the project. So the idea is that at the end of the term, you'll communicate that, like, through either a talk or a written article, something about a research study to the general public. And so, yeah, you get to choose your format. So either an essay or a really short presentation that you'll deliver to us on Zoom. And you have to choose a paper from the literature to be the focus of your project. So that's kind of like the first step, is choosing your topic. And it just has to be something related to social evolution, and it should be an empirical study. So like, the one we read last week is not an empirical study. It's kind of an opinion piece / literature review. So we want you to choose something that's a study, like one that has data. And if you're not sure, if you've got one in mind and you're not sure, we want you to come to me and come to Jenna and ask us, is this a good focus for my project? Okay, so it's worth 25% of your grade. And again, if you have no idea what to cover, talk to us and we can give you some ideas. That was one of like the most fun parts of teaching this class in the past, was like helping people hone in on their project idea. And we can use like the time on Fridays to do that as well. So, although there's actually not much time left, not many like scheduled Friday classes left. Okay, so what is it? So if you do an essay, it's a short article that's about 3 pages single spaced. You can present it double spaced, but that's the kind of length we're going for, with references on top of that length. And what we want you to do is write this as though you're an outside reviewer, and your article explains what the study does, like explains the reason why and why they did it and why it's so interesting, and explains what the authors of the study did, and talks about the strengths and the weaknesses. And we'll have at least 3 references from the primary literature. And there's details about that in the instructions. If you do a presentation, it's really short. It's just a 5-minute presentation because we aren't sure how many of you will choose to do that. Followed by some time for question and answer. And in your presentation, we want you to pretend that you're the author of the paper. So you're presenting the study as though you did it. You don't have to like give yourself their name or whatever, but we want you to take the perspective of the scientist who did the study. And again, like communicate, like, what is the question? Why does it matter? Why is it so interesting? Get us excited about it, and then explain the methods and results. And then we can ask you questions and you can answer them as though you were the author of the study. And so the same idea is that you'll need some references as well. Okay, so what we want you to do after the break week is to submit a proposal. And so this happens after the midterm, but I wanted to talk about it now because it comes kind of fairly quickly on the heels of the midterm. And so the idea for the proposal is that we give you some feedback, and it's only worth 5% of your grade. And the instructions are in that one PDF. And so here's what I would recommend. So start, like, reading week is a good, break week is a good time to do this. Start thinking about the paper that you want to pick. And like one way you could go about it is like think about the lectures we've covered, or look at the titles of the lectures coming up and which ones interest you the most, and just do some Google Scholar searches related to that topic. Maybe if you have an organism that you really like, you could also orient your searches around a topic and an organism. And before you dive too deep, make sure you understand the study, right? You want to pick a study that's one that you will find not too difficult to understand and communicate. And a different strategy, instead of using like a literature search, another way could be to search news articles, right? Because they're like studies that come out more recently will have online news coverage. And that can be a way to kind of hone in on the things that are most interesting to the most number of people. So that's not for everybody, but maybe that will help you find something that really piques your interest. Okay, so in the proposal instructions, there's a series of 7 questions that you have to answer for the proposal, and it's intended to be like a starting point that you would need to make your essay or to make your talk. And you can answer those questions in point form, or if you want to use sentence form, that's fine, but point form is fine. And so my suggestion is to find a paper, start answering those questions, figure out the parts that are a bit murky to you, and then look for additional references that will help build up your understanding, and then kind of edit your answers or update them as you start to understand more. And yeah, okay, so that's all I've got. So at this point, I'm interested if you have any questions. I'll also mention there's no scheduled class the Friday after the midterm. I've left that blank. And what I'll probably do on that day is be available. Like Jenna and I can be available to chat about project ideas. So that's totally optional. But if you have like a paper in mind or you just have no idea, you can like chat with that. That's a scheduled time when you can chat with us. So Kara, did you have a question?
Yeah. Hi, Roz. So I was just wondering for like the presentation, if for corresponding material, if there's anything that you prefer, like PowerPoints or like a potential like poster to go along with it.
Okay, good question. What will you actually have to hand in for the presentation part? I think that all we will need for you from your presentation is the list of references, and we'll be evaluating you on the presentation live. So we'll probably get you to hand in a file with the list of references, but yeah, we won't require you to hand in your PowerPoint.
Okay, perfect.
Yeah, and if everybody, so one of the other things is with the proposal, we have to figure out how many people want to do each type. And, there's some room to switch if you change your mind, but you'll have to let us know, because we'll need to schedule the presentations, and we'll need to figure out, okay, are there few enough of them that we can all attend all of them, or are there so many that they just won't fit in the two like lecture slots? And in that case, like Jenna and I can split up and we can have 2 rooms with kind of parallel presentations. So I want to do the presentations live, but if you're worried, like if you have a poor internet connection, pre-recording it is an option. And so what you could do in that case is pre-record it and like send it to me beforehand if you're worried about your internet cutting out in the middle of the presentation. So yeah, that's a good idea. Thanks, Zainab. And it's really short, but we want you to be live to answer the questions. Like we want you to actually be there. And of course, if somebody like drops off of the Zoom call or something, like we'll find a way to make up for it. So. Okay, any other questions? All right. Okay, so I already found out that this paper people kind of liked, but it maybe wasn't the top favorite. What I have is a bunch of slides about this paper, and then I have a couple of slides at the end where I really wanted to kind of show you the relatedness calculations again, because I feel like those are tricky and hard to explain without a pointer. So, so I just want to say, when I was an undergraduate student, all I wanted to do was experimental evolution. I thought this is the coolest thing I could possibly imagine. You control evolution. Like, how mind-blowing is that? So there's been lots of really famous studies now doing experimental evolution on things like bacteria, fruit flies, things like these moths, where the generation time is short enough that it's feasible to have a lot of generations happen in the lab in kind of a reasonable amount of time. And actually, there's even been experimental evolution studies on mammals, things like mice and like hamsters. But of course, they can't do as many generations with mammal studies. So I have experimental evolution envy. Does anybody want to take a stab at the second question here, just summarizing the main question that the authors of this study were trying to address and their hypothesis? Maybe just the first part of this second question.
I think that they were looking at like their main question would be, does limited dispersal versus like high levels of dispersal tend to select for selfish behaviors, specifically cannibalism. So how does it change rates of cannibalism, I think?
Yes, exactly. So, but a slight, you might have just misspoke, but what did they, okay, so I guess what was their prediction? What of their, did they expect that limited dispersal would favor selfishness, or did they expect that lots of dispersal would favor selfishness?
They said that lots of dispersal would favor selfishness and less dispersal would favor lower rates of
Yeah,
selfishness.
exactly. So sorry, actually, I think you actually said that correctly. I just got confused.
I don't know if I specified.
Okay, thank you, Anna. Okay, so, and I guess I didn't actually answer the question, what is experimental evolution? So it's the idea that we can ask questions about evolution where we manipulate the environment in the lab and we kind of let the organisms have multiple generations in those manipulated environments and test whether the different environments take those populations to different endpoints. So we're not, when you do experimental evolution, you're not doing selection yourself, right? It's different from artificial selection. Artificial selection is where you're like, well, I'm going to breed, like, I'm going to find the most selfish moth larva and breed those and see if I can get a more cannibalistic moth to evolve. That would be artificial selection. Experimental evolution is just, I'm going to alter the environment and just let the moths evolve and see how like environment 1 versus environment 2 influences that trajectory of evolution. And so it's important to have replication. It's important to have, you know, if you have 2 environments in your experiment, it's important to have multiple lines of, or multiple populations in each of your main treatments or your main environment types. And so they had a few of those. I don't remember the exact number. Okay, so just if you have any questions, please just like raise your hand or put them in the chat. Okay, so this was this is their first figure, sorry, this is figure 1, illustrating the environments that Anna nicely described. And these are like the little moth larvae. And so they live in this kind of jelly-like food substrate, and they move their way around. So we're seeing their little like foraging paths as they work their way around and eat and grow before they can like metamorphose into an adult moth. And so, so this is an illustration of lots of dispersal because the larvae are able to move around, and this is limited dispersal. And this figure shows how with limited dispersal, we expect moths from the same family group to end up being close together in space. So like the nearest neighbor of this yellow guy is some other yellow guys, and this like purple female is going to be near other purple females if dispersal is limited. Whereas an environment where dispersal is large, you're going to have a lot more mixing of the family groups. I just wanted to point out, I don't know if people noticed this, something went wrong with the publication of this paper, like they didn't finish this caption. I found that the second last sentence here didn't really make sense. So something went wrong in their editing process. And then the last sentence is just unfinished. Yeah, a few typos. Yeah, it's always like, it's always kind of disappointing when you see that. Okay. Okay, does somebody want to explain figure 2? Oh, Janessa?
I'll try my best.
Yeah.
I read this really late at night. So from what I'm gathering, the hive movement rates are also known as low viscosity because, the hive movement rates are also known as the low viscosity, which means that they're going to be further apart. So there's going to be more cannibalism. And then the low movement is the high viscosity because I guess things are more viscous, so there's not as much movement, and they're going to be closer together, and then that's going to select for less cannibalism in
Right.
short terms.
Exactly, right. So those are the author's predictions. So yeah, you really nicely described how, like, high viscosity means low dispersal. It's like the medium is thicker and harder to move around in. And so this panel shows that that was true. They had these 5 different viscosity levels in their experiment, and then they measured how much the larvae were actually able to move, and they have this kind of plateauing decline where the most viscous food substrate, they were able to move the least, just like we would expect. Yeah, so setting up their like 5 different environments, and then they had some replication within each of those environments.
Yeah, and I also read further that like increasing the viscosity results in more spatial clumping. So there's more interactions between the siblings, and then those alter the social and genetic spatial structures of the population and the system.
Exactly. So their whole prediction all comes down to like Hamilton's rule. And if you're interacting a lot with your kin, we expect that to present a selective pressure against cannibalism. Because if the individuals that you're bumping into and potentially eating are your kin, that's going to be the genes that generate cannibalistic behavior will experience more selection against those genes, because they'll be like destroying other copies of themselves. Yeah. So okay, so how about this question? If anybody is up for answering this. So they maintained the moths for 10 generations. I forget how long that was. It was like a year, maybe. Oh, Neve. Oh, 40 weeks. Okay, great. Thanks, Augusto. Okay, so they just, all they did was kind of let the moths live and reproduce in these environments, and just kind of give them fresh medium every generation. And at the very end of the experiment, they measured this phenotype of interest. I don't know if they actually measure, that raises a good question. Did they measure the phenotype of interest at the beginning? Because you'd also want to know that the lines were all, the phenotype in each line was the same at the beginning of the experiment, but it sounded like they only did that measurement at the very end. So that's a potential issue. But yeah, so they ran them for 10 generations. They used the larva of the final generation to measure this trait of interest, cannibalism. And the figure 3 shows their main result. But the important thing noted here was that they used a common environment. So instead of testing for cannibalism in the environment that these populations had been evolving in, they put them back into like the regular food medium that's kind of probably average viscosity or something, I don't remember. And so why was it important to do that?
I think it's because to make sure that differences in behavior are not in some way linked to the medium. So even though they made sure that the mediums with different viscosities had sort of the same nutrient levels, it's important to make sure that for whatever reason, for example, moths in lower viscosity mediums aren't just having more problems sort of finding nutrients, which could drive them to cannibalism, for example. The common environment at the end shows that the adaptation is really to this dispersal and not to any other factors.
Exactly, yeah, and so Anna, exactly. So they want to show that this is an evolved difference. It's not just an immediate like approximate difference caused by the environment, right? If we tested them in the 5 different environments, then we don't know whether it's an evolved difference between the lines or like an immediate response of, okay, moths in this particular environment just do more cannibalism right away. So that they wanted to establish that it was like an evolved difference between their lines. So yeah, excellent. Okay, so this is the result. And just checking the caption out. It was a while ago that I read the paper. So A and B, it's the same experiment, right? It's just the only difference is the x-axis. So the data on the y-axis is actually the same. But as they showed in figure 1, so figure 1, yeah, the different viscosities were associated with these different dispersal distances. So they're plotting like their main response variable, the amount of cannibalism with respect to the viscosity treatment, but also with respect to how much dispersal actually happened. And figure 3a, gosh, shows this like really beautiful decline that fits their prediction. And it looks like if you look at the relationship in terms of average dispersal distance, it's a bit more, it's maybe a bit noisier, but that there is in both cases a change in the direction they expected. Okay, so here, I think this might be the last question I have here. And it relates to the previous one, but this one's trickier. So they not only tested the final larva in a standard environment, they also reared the parents of those final larvae in a standard environment. And so the idea again is to control for potential confounding variables, like Anna was saying. But can you explain like what exactly it would be? Like what was the issue that they were trying to control for by doing this? They were pretty opaque about it. They just said, oh, it's because of maternal effects. But could you infer, I guess, and explain to like somebody who hadn't read the study why they would want to do that? Why it would be important? Oh, sorry, Neve has her hand up. Maybe from before or now, I'm not sure.
Oops, yeah, it was before, but I can give it a bit of a stab. This one I'm not entirely sure. But would it be in case like either some behavioral traits might come down either epigenetically or possibly learned, I don't know to what extent that would have a role in the larva, but to make sure that, for example, if the parents had a hard time getting food, they wouldn't pass on either genetically or I suppose you could say culturally to their offspring, you know, food is scarce, so cannibalism is all right if it helps you get by, just to make sure that it really is just on this evolutionary level instead.
Yeah, exactly. Right. So the term maternal effects refers to the fact that parts of our phenotype are determined by our parental environment. So, and like there's lots of interest in this for human health, for example, like people who undergo like very stressful times, like nutritional deficiencies and all, and things like that. Though like the signatures of those stresses can be passed on to your offspring through effects like epigenetics. So kind of changes in gene expression in the parents that can be transferred to offspring. Similarly, parents like can endow their offspring with resources. So when a mother lays an egg, she puts resources into that egg, and depending on the environment that the mother experiences, she can sometimes put more or less resources into that egg. So those are proximate causes that can influence all kinds of aspects of our biology, including behavior. So the idea was to really be able to say that this was an evolved difference in the larval predisposition to cannibalism. They wanted to rule out the possibility that it was just like, oh, the mothers were kind of putting more or less resources into their eggs. Like maybe if mothers put less resources into their eggs, their offspring are kind of hungrier or something and do more cannibalism just because of their maternal environment. So yeah, maternal effects refers to kind of the environmental effect, the kind of immediate environmental effect that parents create for their offspring through things like components in the eggs or like hormonal effects when offspring are in the womb, for example. So it really has to do with parental care as the environment. Parental care is part of the offspring environment. Okay, so I hope that makes sense. Do you have any questions about this paper? Oh, okay, I had another question. I thought that was the last one. There's one more. This is another good one. Again, not defined because the paper was kind of written as though you knew a lot of this terminology already. And maybe you would from taking evolution classes. But the question here is, what do they mean by standing variation? And given that the experiment only ran for 10 generations, what do the results really tell us about this behavior in the source population? Niamh, yeah, please do answer if you want to.
So I think standing variation would be sort of just the differences in given behavior or statistics. So in this case, I guess the tendency to cannibalism in a given environment, like even when dispersal is low, so it's not favored, there will still be variation in the different individuals' ability or desire to cannibalize. Sorry, desire would be anthropomorphic, instinct to cannibalize. And these are in fact required for evolution to take place, because it needs these differences to sort of play off of. So given that they observed these differences very quickly in evolutionary time, this would imply that there was quite a bit of variation to start with, so that it could select quite strongly for those who are very prone to cannibalism to begin with.
Exactly. Fantastic answer. Yeah. So in like genetics, standing variation refers to the genetic variation that's already there. And as opposed to variation that comes from new mutations, right? Standing variation is the diversity of alleles that already exist in a population. And so if you're seeing differences after just 10 generations, it's unlikely that there's a lot of new mutations happening that would be causing increases or decreases in cannibalism. And so really what the researchers are showing here in this experiment is that the starting population has genetic variation in cannibalistic tendencies. And by changing the environment, they're able to produce endpoint populations that differed in the prevalence of those initial variants. So the environment in this experiment is filtering on variation that already existed, we think. It's certainly possible that new mutations could arise that would influence cannibalism, but it's unlikely in a pretty small population with so few generations. So, okay, so I think that's the last question. Ah, yeah, that was the last question. Okay, any questions about the paper? Okay, yeah, why were there such outliers? So what are the differences between the red, are the red points the means, and what are the gray points? I don't remember. The red dots are the mean and the line represents the model fit.
I think in the original figures, like when it had the more zoomed up one earlier, it had a whole bunch of gray points showing like all the different data. I think the gray points above and below each mean in this one is just showing like the highest and the lowest value that they got. So it's sort of showing the outliers in the...
Oh, okay. Because yeah, I don't remember because I read the paper a week ago, but for this experiment to work, they would have had like probably thousands of individual larvae in the experiment. And they may have like maybe these dishes or these environments that might have had, you know, hundreds of them and multiple replicates like that within each of the 5 environmental treatments. And so they're maybe they're just showing the extremes, but right. So there'd be more gray data points that just aren't being shown for some reason. I don't know why. It doesn't seem like it would be that hard to show them. Oh, Augusto, yeah.
Hi. I was just a little confused about figure 2 and on what they were actually trying to show. Yeah, on the right side with all the arrows and whatnot. Because yeah.
Okay. So yeah, I'm not a huge fan of this one either. Because I see. Okay, there's actually a difference in the way that larvae are clustered here. Okay, So the rows, they've only shown 2 rows, but you could imagine that there's like 5 rows here that are supposed to represent the 5 levels of viscosity. They're 5 different environments. And this is the highest viscosity. So the larvae are really clumped. And this is the lowest viscosity. So the larvae are able to move freely and mix with other non-kin. So it's kind of weird that there's 7 arrows because they've got 5 levels. This is supposed to be the top level,
Right.
level. Yeah. And then the columns are showing you evolutionary time. So like, here's the beginning of the experiment, and this is supposed to represent the end of the experiment. And so like the x-axis is supposed to represent 40 weeks. And so every week they'd be transferred to the population would be like a given box would be transferred to a new fresh food box and to do another generation. And I guess within each of these 5 environments, they had 3 replicates. So they had like 15 boxes at any one time. And within each box, there's probably, I don't know, 100 larvae. I don't know if they told that information. I just don't remember. But it's actually not a huge population size for an experimental evolution study, like unless they had thousands and thousands of larvae in each box. It's a pretty small population.
Yeah. Oh yeah, it makes so much sense now. I thought somehow they were changing the larvae within the viscosity levels, which didn't make much sense, but I think I get it now. Thank you.
Okay, yeah. Any other questions? Oops, went too far. Was this surprising to you? I was thinking about it this morning and I was kind of thinking, well, of course, this is what they found. But I think it's often like easy to think that after the fact, especially when the results are so clear. All right, if there aren't any other questions, I'll go on. Oh, another. They expected, oh, interesting. So somebody who was not in science was able to come up with the same prediction from this study. That's interesting. That people, I mean, it does make sense that people should have an intuition that you like it would be maladaptive for animals to eat their kin. Okay, so that's all I have for the paper, but I just wanted to kind of go over these slides again because when I recorded them this week, I can't use my pointer when I take the recordings in Keynote, so I thought it would be helpful for me to kind of show this. And so I will say, I'm sure that there'll be questions on the midterm that have to do with interpreting like relatedness scenarios. I'm not going to give you really, really tricky ones. I'm going to focus on some of the obvious ones. And it's easy to look up what the relatednesses are for some standard relationships. Like there's a table in the lecture that shows you a bunch of kind of simple familial relationships and what the relatedness value is. But I just thought if you want to know where those come from, I could show you that. But for the really complicated ones, it becomes impossible to kind of answer complicated ones using the methods I'm going to show you. And in that case, like there's software packages and things like that where people can estimate relatednesses. And so some of the answers that I put on the third slide came from like, there's no way to solve it with simple methods. You have to, for me, I have to use the software. But these ones you can solve with these simple methods by hand. So if you know that A and B are siblings, the way you calculate the proportion of their genome that they share is by first saying how many common ancestors do they have, like at the kind of highest level, and they have 2 if they're siblings. Like two direct common ancestors by descent. And so we're going to add up 2 numbers, one for the amount of genes shared through dad and one for the amount of genes shared through mom. And that's my 2 probabilities here. And to get each probability, I just go from A to B or from B to A, and I count up how many paths I take to go up to the shared ancestor and then to the other individual. So in this case, to go through dad, from A to B through dad, it's 2 steps. And to go from A to B through mom, it's 2 steps. And so for every step, you just like take 0.5. So every direct descent is worth a gene sharing probability of 0.5. And you just raise it to the power of how many of those steps you have to take. So the calculation ends up being 0.5 to the power of 2 steps for genes shared through mom and 0.5 to the power of 2 steps for genes shared through dad. And that ends up being 0.5. So on average, full siblings share half of their genome. Now, of course, if those full siblings are identical twins, who can tell me how much of their genome identical twins share?
It'd be 100%, right? Excluding like de novo mutations.
Exactly, yeah. So R for identical twins is 1. It can't be any higher. They're clones of each other. If the twins are fraternal, so that means that they arose from 2 different eggs, but they shared the same womb, the same gestation. What would the R be for fraternal twins? Fraternal twins come from 2 different eggs and 2 different sperm.
Also 0.5.
Exactly. Yeah. And thanks, Flora, too. Exactly. Okay. So, so half sibs, now they just share 1 common ancestor. So we just have 1 probability in our sum, and we just count up the number of paths and we raise 0.5 to that power. And so that's why we get 0.25. So is this making sense? I didn't want to like belabor it, but I feel like it can be confusing. Okay, good. Yeah, it's the kind of thing where it's best if we have like a whiteboard to really point to what's happening. Okay, so for grandparents, it's the same idea. So if I want to know how much is grandson related to grandma, or equivalently grandpa would be the same, we just have to say, okay, how many black paths do we go through to get from A to B or B to A? It's the same, right? Grandma's relatedness to grandson is the same as grandson's relatedness to grandma. And so we've got 1 path, 2 paths, so it's 0.5 squared. And because this is like B is a direct descendant of A, that's just the path we have to consider. And then here's where they get really tricky. This is still one that you can do by hand, but it's confusing. So these are like cousins, but they're like very close cousins because their parents are 2 sets of siblings. And so normal cousins share 1 set of grandparents, but these special cousins share both of their sets of grandparents. They have 2 sets of grandparents in common. And so they've got 4 shared highest level ancestors. And so we have to add up 4 numbers, and we have to like find our way from A to B through like all possible ways through each of the ancestors. So if I look at this grandpa, I have to get how many ways can I get from A to B through that grandpa? And it turns out that there's 4 steps to get. Yeah, so sorry, 1 step, 2 step, 3 step, 4 step. So that's where that 0.5 to the power of 4 comes from. And then I have to repeat that for all of the other 3 shared ancestors. So I end up getting, and it ends up being the same, right? It's 0.5 to the power of 4 added together 4 times. And so that's where this comes from. So I'm not going to give you like a really tricky one, but I'm going to make you think about how relatedness plugs into Hamilton's rule. And I'll probably ask you to consider some fairly simple ones. But because we're not like practicing this a lot, I'm not going to get you to answer this. But the point is, regular cousins, they just share one pair of grandparents. Regular cousins have just a relatedness of about 12.5, but these are, even though there's no incest here, these cousins are going to be share a lot of their genome. Yeah. So we read a really interesting paper last time we taught the course that I didn't have us read this time, but I might show it in one of the lectures after the midterm, that talks about how different human cultures through history have had different kind of rules about who's allowed to marry whom, and that this can influence the relatedness structure of human populations, and how that might in turn influence our cultures. And so the researchers did something really ambitious where they measured all kinds of aspects of like cooperation and selfishness across human cultures around the world, and then correlated that with the different cultures' rules about kinship marriage. So it's a very like surprising and ambitious paper that was super interesting to read. But it made me wonder about these more complicated scenarios, and especially cousin marriage, because cousin marriage is something that's actually been quite common throughout human history, and it's of course common in animals too. But those ones are tricky to calculate, so that's why I don't have a graphic for them. I had to like use software to figure it out. Okay, that's all I've got. Are there any more questions? All right, not hearing any. I'm going to stop recording.